Hi everybody, welcome to another Chats with Chaudhry. I hope you're safe and well. It's a lovely day here in Surrey, and I'm delighted to be joined by Melissa Sherman, who is CEO of MOBILion. I had to get it right because I was going to call it MOBILion, but this MOBILion Systems. And today we're going to be learning about SLIM technology and hear about a new product launch, which we'll talk about shortly. So Melissa, how are you?
I'm great. Thank you so much for having me with you today. I've been looking forward to it.
Well, it's lovely to see you. In fact, we were just discussing beforehand, actually, viewers, that the last time I saw Melissa was almost this time last year to the day, although it was still virtual, because ASMS had gone virtual, and you were having a press conference then, weren't you, if I remember correctly?
We were, yes. We were launching MOBILion and what's to come in MOBILion's future last year at virtual ASMS.
Right. Well, perfect segue to my first question to you, actually. So there you go. So for those people not familiar with MOBILion, would you mind giving them a very quick overview of the business and also a little bit about your own background as well, if you don't mind?
Sure, yeah. So I'm the CEO of MOBILion Systems, Inc. What we do, about MOBILion. So we're an analytical instrumentation company commercializing a best-in-class ion mobility separations technology that, when integrated with high-resolution mass analysis, provides unprecedented speed and resolution to separate and identify molecules that other instruments fail to detect. So we say at MOBILion, if you remember nothing else, we reveal what others leave unseen. So that's we reveal what others leave unseen. And from a customer and applications perspective, we help our biopharma customers bring drugs to market faster, and we help academic and clinical researchers accelerate biomarker discovery and validation. And maybe going into a little bit about the technology and where our company came from, where our foundation started.
Oh.
So our products are based on the SLIM technology, as you said, that was developed at Pacific Northwest National Lab by Dr. Richard Smith. So those that have been following in the ASMS community, Dick Smith has been talking about the SLIM technology, oh gosh, for probably about 6 or 7 years now. And MOBILion has exclusive license to bring it to market. And we set out, our history goes back to 2017. We were established then when we hired our first employees, set up our headquarters in the suburbs of Philadelphia, and then set out to transfer the technology from Dick's lab in Washington State to our headquarters here in Philly. 2019, we began our product development. Our first product is a product development collaboration with Agilent Technologies. In 2020, in spite of COVID and the pandemic, we beta launched our product. So the product that we're debuting and launching in a couple of weeks here, we beta launched last year with a handful of beta users. And then now, like I said, next month on June 15th, we're virtually launching our product to the community. So everyone will get a glimpse of it then.
All right. So this is really a bit of a teaser for people then, really, isn't it? So let's talk about your upcoming product. I understand it's called I might get this wrong, HRIM-MS, or is there another sexier term for it, or is that what it's called?
So yeah, good question. We're not debuting the official name until June 15th. So you have to tune in. If you want to participate and tune into the event, you can find info about it on our LinkedIn page and Twitter. But yes, the platform we call HRIM mass spec, and it's high resolution ion mobility mass spectrometry. Our first product is integrated with Agilent 6500 series. So it's the highest resolution separations, highest resolution mass analysis to, again, reveal what others leave unseen and be able to separate and identify molecules with unprecedented speed and resolution. So the official name, you'll find out on the 15th. It's going to be a lot of fun.
Fantastic. What a true marketeer you are, Melissa, I have to say. So, so in terms of workflows and applications and also helping researchers, what are sort of workflows and research challenges that you're hoping to address with the HRIM-MS?
Yeah, it's a good question. So we are targeting biopharma characterization, so specifically released glycan analysis, intact subunit, peptide monitoring, and then also in kind of the omics space, targeted glycomics and lipidomics. So really focusing where they're complex, really challenging analytical systems, or analytes, I should say. So in glycans and lipids, there's kind of a threshold of resolution because there's so many either isomers or just minor differences between different molecules that are very difficult to separate and identify. So that's really where we focus, where there is a deficiency in resolution, where you really need that high resolution separation, or where you need speed. So, that's the other aspect of it. I'll give one example, some work that we did at Janssen that we actually talked about last year at virtual ASMS. And this was looking at glycans, and we compared what is the incumbent gold standard, which is Hillick liquid chromatography. Yeah. And Janssen looked at it as I think 20-something glycans in the sample. And in a 15-minute Hillick liquid chromatography analysis time, they could see 53% of the glycans of interest, where when we were using our high-resolution ionmobility separations, we could see 100% of the glycans in a 2-minute analysis time. So it's kind of that more complete coverage with very quick analysis times. We've also we've partnered with Agilent on the mass spec, and we've also partnered with Protein Metrics on the data processing software. So we're offering a complete expedited and efficient workflow in Protein Metrics by providing their simple automated data analysis capability, particularly for our biopharma customers.
Fantastic. Now, you may have partly answered this, or maybe answered all of it, but so how does HRIM-MS differ from other ionmobility instruments on the market, and which applications is it really used for so far?
Yeah, so let me talk about the applications first. So I gave the example in glycans. I can give an example also in peptide monitoring. So again, biologics characterization and, kind of following the mega trend in the pharmaceutical industry of small molecule to large molecule therapeutics. And with these more complex protein-based therapeutics, there can be characterization challenges, and it requires higher order instruments to be able to adequately characterize these molecules. And so that's really where we're focusing, is predominantly in the biopharma market, which is what makes it important and meaningful that we've partnered with Protein Metrics, because they have kind of, a best-in-class automated data analysis program for biopharmaceutical customers, but then also in academic and clinical research in omics by being able to provide 2 to 5 minute analyses of samples, where with LC it might take 30, 60, or 90 minutes. A specific example and workflow is in peptide monitoring. So again, with what is LC mass spec today, it's typically a 60 to 90 minute analysis time. We just published an article in JASMIS, I think last month, that shows our high resolution ionmobility confirms protein identity with greater than 95% sequence coverage in either a 2-minute injection to injection runtime or a 20-minute injection to injection runtime. And so what we show is it's not only the speed, but it's the additional information, again, revealing what others leave unseen. In that work, where there's looking at the chromatogram and there's one LC peak, we can pull that apart with our high resolution ionmobility and see that there are 2 clearly resolved peaks at that same mass to charge under that single chromatographic peak. And so what it demonstrates is that we can analyze, or we can elucidate, I should say, in this case, in example in the publication, deamidations that were missed with LC alone. So for our pharmaceutical customers, it de-risks under reporting post-translational modifications. And then in addition to that, provides significantly greater sample throughput per instrument. So there's a workflow advantage as well.
Fantastic. So now, sorry, you can say
how it differs. Yeah, I don't think I answered how it differs from other ionmobility. So the form factor differs. So I don't know if you can see this as my little visual aid here. So the form factor, the inside guts of the SLIM technology, is printed circuit board based. And this is actually an example of what the exact kind of inside looks like with the electrode pattern. And so that form factor, because it's printed circuit board based, it's easier to manufacture and it's more tunable. So we can change, it's really kind of, if you think about it, a smart technology in that we can change the electrode pattern, we can change the electronics, We can tell it to do different things. We can send certain ions to the detector, certain ions to the trash can. So the sky's the limit in terms of our product development potential. And the product that we're launching in 2 weeks from now is the first product. And the first product was meant to be simple, right? Keep it simple, stupid with your first product. And it's a 13-meter or 42-foot separation path length condensed in a component about the size of a briefcase. And the reason we can get such long path lengths is because Dick and his team figured out with this form factor how to turn these ions around corners on these electrodes without them being neutralized or lost. So the SLIM, structures for lossless ion manipulation, is real. The ions are not lost inside the chamber, so you preserve sensitivity. And then because we have this serpentine path length, the degree to which you can separate is proportional to the length that you're separating. Right. So we have these super, super long resolution path lengths condensed into a really small viable form factor. And so compared to other eye mobility technologies, because we have this really long path length, we get higher resolution, revealing what others leave unseen. And then it's simpler. It's single pass. We don't have to do loops. We can do a single pass, 13 meters, which is more simple, even from a data processing perspective. We preserve the sensitivity. And the other final point, which is really important, is we also don't have to narrow in on a mass range. With some other technologies, you have to just narrow in and focus on a certain M over Z range. We have the broadest M over Z range, and we put all the ions through at the same time, and we're able to still hold that ion capacity and adequately separate them.
That's really interesting. Thanks for sharing that. So I know that HRIM-MS has been used for research in SARS-CoV-2. So can you talk about some of the outcomes that you've had in that particular research?
Yes, we have. So when we think about COVID, right, much has been said about the glycosylated spike proteins on the capsid of the COVID. And right, we've all seen pictures of it now, and we know that those spikes are critically important. And it turns out that the glycans tethered to the proteins of that spike are important to how COVID's mechanism of action and in terms of developing vaccines and therapeutics against it. So we have a couple collaborations. One is at CCRC, the Complex Carbohydrate Research Center at the University of Georgia, where they're doing some research in looking at glycopeptides of the COVID spike protein. And then also we're working with Oxford University. So Dr. Rod Chalk is profiling the glycopeptides associated with the SARS-CoV-2, which will be used to add another dimension of characterization data to an open access database that a group of researchers are developing.
Fantastic. So you mentioned a bit earlier that obviously you've got this launch in a couple of weeks' time, and this is the first of hopefully many, and there is lots of opportunity with the technology that you've developed. So what are your plans following the launch into 2022, and what do you see the future really for MOBILion Systems, and also the technology, more importantly?
Yeah, it's a good question. I mean, you know, right now we're laser focused on the launch, and our primary goal with the launch is providing best-in-class customer service for our customers. And, you know, when you launch a first product and the first technology and the first time it's on the market, is everything going to go perfectly? We sure hope so, and we strive to, but, you know, it might not, and there might be issues. And so our goal really as we launch this first product and bring this technology finally to the masses is really to be best in class in terms of customer satisfaction and provide VIP concierge service to our early adopters and our first purchasers of the product. And we're thrilled to be working with Agilent and Protein Metrics in kind of lockstep, right? Because we all are, it's an integrated system that we are looking forward to keeping our mutual customers happy. So we're really focused on the launch, really focused on working with the customers that are our early adopters. And then beyond that, we've got a whole portfolio of products. Obviously, I can't talk a lot about it, but we do have confidential collaborations in the works to integrate with other mass spec platforms. And then we also look at the value chains in which we're playing. So we have talked publicly before about this mass spec integrated products. So we have our HRIM product that integrates with Agilent's mass spec as either a field upgrade if our customer has the appropriate model of mass spec, or as a full system. That's really for late development R&D applications. And the high resolution separations and high resolution mass analysis are appropriate when you want to see everything and you want to know everything that's there so that you can identify what molecules are important, for example, in biologic characterization. But once that characterization or those critical quality attributes that have been identified move downstream in the value chain, say in manufacturing or in a quality control lab, you don't really need to see everything of everything. You just kind of need to know what you're looking for, what you're interested in, that peptide map, that glycan profile, whether it's there or not. And so we've got a complementary product that's our Gen 2 product portfolio that is an all MOBILion, all fully integrated product that's even easier to use, push the buttons, get the result that you need, all the components under one hood in a sleek design that's more appropriate for a less sophisticated operator downstream in the value chain of biopharma. So that's our Gen 2 product concept that's coming next. And what it does is, if the pharmaceutical researcher says, okay, I'm developing this assay, then they're not afraid to develop something that they can't translate downstream. They can translate it downstream because it's that same slim technology at its core and in its guts, so that it makes it amenable to translate that method downstream.
And do you think, because looking at the technology, I mean, right now you're focusing very much on pharma and biopharma, but do you think it's also applicable then to food and drink, chemicals and other industry segments, apart from, because obviously they're less regulated, some of them, and also the analysis may not be required to be as in-depth as you would have necessarily in biopharma, as you mentioned earlier?
Right, definitely. And we have exclusive access to the technology for all applications. So for us as a little company starting up, we had to focus, and we chose to focus first on biopharm.
Sure.
But yes, there are many other applications where speed, resolution, ease of use, efficiency, simplicity would be beneficial to a variety of different workflows in a variety of different industries.
Fantastic. So if we want to get more information, apart from obviously watching this wonderful video and going to your website and going hopefully to the press launch if they can, or in your product launch, where can they get more information about MOBILion and also the technology we've talked about today?
Yeah, so our website, the easiest, www.mobilionsystems.com. You can follow us on Twitter at MOBILion Systems. I would say we're probably the most current and most active on LinkedIn. So we've got a number of webinars coming up. The big launch, like I said, June 7th is our team that, has been working so hard for 4 years to bring this product to market. That will be on June 7th as an internal and VIP event. And then on June 15th is the official virtual launch. And if anyone wants to attend, they can follow our LinkedIn or go to Twitter and get those details and register.
Fantastic. Well, you are very active as a company, and you personally are, I know, because I follow you on LinkedIn as well. You speak at a lot of events as well. I know that, and doing a lot of seminars and workshops as well. So, Melissa,
likewise. I was going to ask if you ever sleep for as long, because I don't think you do.
Well, I don't cover India. That's the thing. I don't cover the subcontinent. I cover America, North and Europe, so that I have got some time to get to sleep. But this is about my 170th interview since May last year.
Wow,
so they've gone very well, very well, and I'm delighted finally to
get to. You do a great job. Yeah, you do a great job. I feel like I'm on with a celebrity. I know. Likewise. I follow your celebrity status, and I'm really honored to be here.
No, you've been too kind. But thank you very much for sharing the information and sparing the time. I know you're very, very busy. So thank you very much for doing that. Good luck with the launch. Hopefully, I'll be either at the VIP one or at the press one, and looking at that as well. But it sounds really exciting stuff. I know genuinely, listening to your presentation a year ago, and I'm not just saying that to flatter you, but it was a really interesting presentation you did at SMS last year. The technology sounds really exciting, particularly when you can see it sort of fits into such a small footprint there, and it gives you lots of flexibility. So it's a fantastic bit of technology. So thank you very much. Good luck with the launch, and lots of success for the rest of the year and next year as well.
Thank you. We'll see you soon.
See you soon. And thank you all for watching. If you've got any questions for Melissa, feel free to put them in the comments below the video, or message her directly on LinkedIn, or follow the LinkedIn pages. As I said, they are very, very active on LinkedIn, so it's definitely worth following the MOBILion page. And also check out the website. The link again will be above there. And I will add the link to the official public launch above the video as well, so that if you want to see that, then the details will be above the video as well. So until then, until next time, stay well and stay safe. Bye-bye.
Bye.